Welcome to week 2 of Chemistry 128, Introduction to Chemical Biology. I'm Professor Weiss. I'll be talking to you today about reactivity. Okay, so last week we talked about the molecules that compose your cells, and our goal this week is to understand how those molecules interact with each other. There are two forms of this interaction. The first kind is that the molecules can decide to react with each other. They can start to form covalent bonds. Bonds can break, bonds can form. So we want to understand this property that we're going to call reactivity. And to understand this, we're going to look at arrows and the language of arrows, which organic chemists have developed as a way of communicating this reactivity. I have to tell you, I think that this is one of the great achievements of organic chemistry. This is one of those accomplishments that all humans can be proud of, because it reduces something that otherwise seems mysterious to a simple set of rules from which you can derive many reactions, essentially all reactions found on our planet. And to me, that's really exciting, because that means that this language is universal, and it's one that's very broadly applicable. And so that's my bias going in, is that I think this is really cool. Okay, so we're going to have a quick review of arrow pushing, and then I'm going to show you examples of applying this language of arrow pushing and this language of reactivity in chemistry to the chemistry that's found on our planet before life started. This is a type of chemistry called prebiotic chemistry. Now, obviously, there were no humans present to observe directly what was going on. However, we can infer what was going on in this prebiotic period, and this is an artist's conception of what the planet might have looked like from both a fossil record and also from experiments that attempt to recreate the conditions that were found during that prebiotic period. Okay, so we're going to be using what we learned to really look at synthesis of the molecules that compose a cell. And then the next topic we'll talk about this week is making molecules using a combinatorial approach. This is essential in chemical biology. This combinatorial approach takes place in your cells. It's one of the reasons why your immune system can very rapidly respond to foreign invaders. And it also is used in many chemical biology laboratories around the world. And so for this reason, I have to introduce this concept of combinatorial chemistry and combinatorial biology to you this week. And then finally, we'll look at the second mode of molecules interacting with each other. Recall at the start of this, I said there were two modes. The first mode being reactivity, that forms covalent reactions that results in covalent changes, bonds forming and breaking. The second mode is non-covalent interactions. This is when two molecules slide alongside each other and decide to form a complex with each other. And the rules that determine whether or not this complex forms are also rules that we can understand. And importantly, this is also a really tough frontier for chemical biology. So while I'll be able to tell you about the rules for reactivity and covalent bond breaking and bond forming reactions, I cannot reply with such certainty when we start talking about non-covalent interactions. There's a lot less that we understand, and that makes it one of the challenges. But at the same time, it also makes it really exciting, because that means there's opportunities for people like yourself to get out and do new experiments to start to elucidate those types of rules. Okay, so I have some announcements before we go in. That's kind of the overview. Let's zoom down and look at the particulars. First, for this week, I'd like you to read Chapter 2 in the textbook. That's this book here. Now, there's occasional times where the treatment in the textbook is more advanced than what I'm talking to you about. For example, there's information about inversion of phosphate geometry, of phosphorus geometry. I'm not going to discuss that. And if I don't discuss it in the lecture, then don't get too hung up on it in the book. Okay, so simply skim the concepts that are not presented in the lecture. Okay, so if I don't talk about it in lecture, it's not important for the class in terms of our exams and what I'll be testing you on. Simply skim through it. Homework. I'd like you to work Chapter 2 problems, in particular, every odd problem. And there will be a worksheet to guide our discussions this week, which will be posted to the class website. In addition, there will be one handout this week which will be posted to the course website. Please download this on Tuesday, skim through it. This handout is an example of a journal article report, which I used to call the book report. And then on Thursday, I'll discuss it with you in further detail. Okay? At this point, I would usually ask if you have any questions. If you do have questions, then you can either email me or the TAs. Okay, so let's review where we've been, and then we'll get started on what I told you about at the beginning as our big picture. So we want to understand the function of human cells at the level of atoms and bonds. This is the smallest unit that actually is meaningful to us as chemists. And as I described to you last week, cells are bags of molecules. They are bags that are chock full of molecules. The molecules are stuffed inside the cells. There is no elbow room. These things are jam-packed into the cell. So because of that, we expect lots and lots of interactions, which is our topic for this week. But I'm getting ahead of myself. Let me continue reviewing what we talked about in the previous week. First, we talked about the composition of a gene as an on-off switch with instructions. We talked about how molecules are synthesized in the cell using the template of DNA to a messenger RNA, which then is translated into proteins, and then proteins and RNA carry out all the various instructions that are articulated by the DNA. We also discussed 6 types of organisms, but in this class, we're going to be generally talking about either bacteria or human cells. Okay, and it turns out there's a lot of chemistry in just bacteria or human cells. So our goal in this week is to reduce the complexity of diagrams like this down into a few rules that chemists like ourselves could understand. Okay, so let's get started with what is life? What is the stuff? What are the molecules that compose cells? What are the rules that govern them? In 1948, the physicist Erwin Schrödinger wrote a very influential book called What is Life? I highly recommend this book to you. It's a slim little volume, and it's a fun read. It's not particularly challenging, but the concepts that he presents to me are really earth-shattering. These are paradigm changing. What Schrödinger argued is that the molecules that govern your cells, that allow organisms like yeast and bacteria and humans, that allow organisms to live, those molecules are governed by physical laws, by the same laws that we talk about in chemistry and physics classes. There's nothing special or unique about the molecules found in living organisms. They are simply molecules that are governed, again, by physical laws. So this book persuaded a generation of physicists to explore biology after World War II. This was an amazingly influential book. And this persuaded this generation that include great scientists like Francis Crick and Jim Watson and many others to explore biology, and to do this by applying concepts from physics and concepts from chemistry. And the results are more or less what I presented to you last week when we talked a little bit about the structure of molecules. So this is good news for us. The good news is everything that you've been learning about in chemistry classes before now applies to biology. There's nothing special about biology. There's no sort of life force that animates molecules found inside the cell. No, rather, the same rules that you learned about in general chemistry, that you learned in organic chemistry, those apply to the molecules found inside your cell. Okay, so let's talk a little bit about those molecules found inside your cells. So our goal is to understand first the reactivity of those molecules, and then second, we'll talk about their non-covalent interactions. So covalent interactions, reactivity first. In organic chemistry, you learned the powerful language of arrows, which are a way of depicting the overlap of molecular orbitals. Let me remind you of some conventions of those arrows and the conventions of this language of organic chemistry. So the first of these is that these arrows depict the overlap of molecular orbitals so that they show, for example, electrons in a highest occupied molecular orbital overlapping with the unoccupied lowest energy molecular orbital of the second reactant of the reaction. Okay, so in this basic reaction, we have an amine, we have a ketone, and the two of these are going to be reacting with each other. So if you take amine and you take ketone and you mix them together, we can predict in advance that a reaction will take place. And here's why. What we can predict is that the lone pair on the nitrogen is going to be highly reactive. Why is that? What's special about that lone pair? It happens to be very high in energy. It is a highest occupied molecular orbital. And it's going to want to react with the pi bond, this carbonyl functionality of the ketone. What's so special about the carbonyl functionality of ketone? Well, it happens to have a low energy, unoccupied molecular orbital. Okay, now let's break down what these molecular orbitals actually look like. What this looks like is the lone pair of this nitrogen on this nitrogen is found in an n orbital. So it's in a high energy state. It's the highest occupied molecular orbital, the HOMO, and it's going to be overlapping with the lowest energy molecular orbital of the carbonyl of the ketone, which happens to be the anti-bonding orbital of the pi bond. Okay, this is what it looks like in terms of molecular orbitals, and this is what it looks like on top in terms of organic chemistry and how organic chemists speak. Good news, we organic chemists have agreed to the convention that we will depict complicated reactions like this one using this simplified descriptor. Okay, and this is good news. I don't think anyone wants to spend lots of time on the test deriving what these molecular orbitals look like and trying to describe an antibonding orbital in terms of the lobes and so on and so forth. It would be just way too complicated. So we're going to be using this description here. Now, the real challenge for us comes from the fact that the molecules that we talk about in biology oftentimes have multiple functional groups. It's not atypical for a biomolecule to have, say, hundreds, if not thousands of carbonyls, or to have thousands upon thousands of different lone pairs. So the real challenge is for us to figure out which of those lone pairs and which of these carbonyls is actually going to engage in a reaction. And when that happens, we're going to fall back on orbitals to decide which of these is going to be most reactive. Okay, so again, what we're going to be talking about is this overlap of molecular orbitals. That overlap of molecular orbitals, the filled-unfilled overlap, leads to the formation of new bonds and consequent breakage of others. Okay, so when this lone pair overlaps with the antibonding orbital of the carbonyl, the pi star orbital of the carbonyl, the result is a new covalent bond directed by this first arrow. Now, on the other hand, we know that this carbon can't have more than 5 bonds to it, or can't have more than 4 bonds to it, and so 5 bonds would be disallowed. And so for this reason, in concert with this formation of a new bond, there's breakage of the pi bond between carbon and oxygen of this ketone. This is good news, right? This totally makes sense, because what we're doing is we're populating this antibonding orbital, and in doing so, we're making that pi bond break, right? If you put electrons into an antibonding orbital, what does it do? The bond breaks, hence the name antibonding. Okay, so this overlap to me is kind of like the peanut butter and jelly of organic chemistry. We're always going to be talking about a HOMO, a highest occupied molecular orbital, overlapping with the lowest unoccupied molecular orbital. And in the same way that peanut butter and jelly taste so good together, orbital overlap works so well. It is so complementary in terms of reactivity. Okay, so let's get back to our challenge again. The challenge is in biology, we oftentimes have many different possible reactivities. We oftentimes have many different possible reaction mechanisms that we can draw. Despite that plethora of possibilities, what we will see is that there is oftentimes one and only one true mechanism, dominant mechanism for a particular set of molecules. And again, this is good news. Okay, so for example, let me show you sort of an easy case where we're going to be looking at reactions, two possible mechanisms, one that makes chemical sense and one that does not. And so by doing this, we can start to eliminate a lot of different examples. Okay, so here is a clash of two possible wills. In this reaction, one possible mechanism has the lone pair attacking the antibonding orbital of the carbonyl and going through the transition state that's depicted down here. This reaction is an addition-elimination reaction. It goes through this transition state in addition, and then in the elimination reaction, the chloride is eliminated, giving us a substitution of nucleophile in place of chlorine. Okay? Makes sense, fundamental reaction. A different type of reaction mechanism might look like this, where the nucleophile directly displaces the chloride. In doing so, the nucleophile, lone pair on the nucleophile, is populating the sigma star antibonding orbital of the bond between the carbon and the chlorine. Okay? So two possible mechanisms. One involves the pi star orbitals. This one involves the sigma star orbitals. And I guess at first blush, these two reaction mechanisms might both look totally legitimate and both equally valid. The problem is they aren't. That we can actually readily eliminate the reaction mechanism on the right that is governed by the SN2 reaction. Instead, what we can do is actually very quickly decide that only the addition-elimination reaction will work. So returning to this clash of two wills, why don't we look at a transition state or a reaction coordinate diagram for the two possibilities, which I think tells us which possibility is correct and which one is wrong. Okay, so this reaction coordinate diagram is depicted over here. So in one reaction that I showed on the previous slide, the mechanism is an SN2 reaction, and on the right, this is the addition-elimination reaction. Okay, so in this, I acknowledge this is a complicated diagram. Bear with me. So over here, these are the starting materials. This is the acid chloride. This is the nucleophile. And again, if this reacts through an SN2 reaction, you will get this left reaction coordinate. And if it reacts through an addition-elimination reaction, you get the right coordinate. Now, two possibilities, small little hill, big hill. Which of these two is preferred? Small hill, big hill. All right, now let's just imagine. You're an electron. You have to decide. Which one would you prefer? Would you prefer tramping up the very steep ski slope, or would you prefer the much shorter hill? Okay, I will tell you also that electrons are lazy, that they do not expend any extra energy than they need. And in doing so, they are going to prefer very strongly the tiny little hill, or the much smaller hill, of the addition-elimination reaction to the SN2 reaction. Okay? This makes sense. That's the way electrons live their lives. So what this tells us is that, yes, there are two possible reaction mechanisms for this reaction, yet only one is actually correct. The only one that's correct is this one on the right, the addition-elimination reaction. The one on the left has to go through a much higher energy SN2 reaction. Okay, now I'm going to explain in greater detail in a moment why it is that the one on the right is preferred than the one on the left. Okay? To understand that, I need to tell you about three possible components of orbital overlap. So the energy in this interaction is proportional to three components. Okay, and let me go back. Recall over here that in reaction coordinate diagrams, the y-axis depicts energy, where a higher number up here indicates higher in energy. And again, electrons being lazy prefer lower energy. Okay, so that again is why the smaller hill is preferred to the bigger hill in terms of which side to go on, left side or right side. Okay, now this energy is proportional to three components. Component number one are charge-charge interactions. Okay, so if these molecules happen to have plus charges and minus charges, then that will have some interaction, some Coulombic interaction. In addition, if the molecules have a repulsive interaction with each other, that will also contribute energy as well. Okay, so charge-charge interactions, these are governed by the social convention like opposites attract. Okay, so in social circles, opposites attract, I think, is commonly accepted. It works as a formula for dating websites. It also works reasonably well as a formula for molecules as well. So happily, social conventions mirror atomic formulas. Okay, so charge interactions are one possibility. If I go back, you could see that we don't really have any charge interactions operative in this mechanism as depicted here. Nucleophile is neutral, acid chloride also neutral. Charge interactions off the table. Second term, repulsive interactions. So this would be if the molecules have some sort of steric hindrance that prevents them from overlapping with each other. And this is a really important component in terms of preventing molecules from interacting. It's used extensively in biology. It's used extensively in enzymatic catalysis. Again, over here, that doesn't seem to be a possibility, right? The nucleophile has a wide open lone pair. The acid chloride, similarly wide open. It has just a methyl group on this attached to it. So there's really no repulsive interactions that are operative here. And by the way, just to remind you, the repulsive term of this equation is the term that allows this hammer to pound in the nail in this wall. Okay, so these repulsive interactions, that's basically the Pauli exclusion principle. That means that electrons cannot occupy, that more than two electrons cannot occupy the same molecular orbital. Okay, and so for this reason, hammer starts pounding on nail, nail goes into the wall to get away from hammer. Okay, they don't, suddenly merge with each other and magically start to create some sort of hybrid material. Okay, things don't happen that way. Okay, so repulsive interaction is clearly important, not so operative in this reaction, right? These two can snuggle up as close as they want. There's no, prevention of that by, steric shrubbery. Last one, attractive interactions. This third term I would describe as mysterious, right? This is not the term that we're used to talking about. This attractive interaction is nothing more than the field-unfilled overlap that I've been talking to you about today. Okay, so here reduced down to its terms is a different representation of the same equation 1 from up above. In other words, the reaction energy for a particular set of interactions is proportional to Coulomb's law, which governs charge-charge interactions, plus the steric terms, minus the field-unfilled orbital overlap. Okay, and it's this third term over here that governs whether or not the molecules actually get to form and to break bonds. Okay, now, here's the deal. The problem is that these three terms interact in a complicated way. Okay, that if we go out and just start applying this equation to every possible social situation we find ourselves in, we're going to have trouble. Okay, and I guess the most obvious thing is the opposites attract rule only carries you so far. Okay, before you get married to your snugly significant someone, it might be a good idea to find out whether that opposites attract thing carries over to I don't know, temperature of the bedroom or something like that. Okay, so for this reason, this equation over here is a good deal more complicated. Why don't we take a look? Okay, so opposites attract. Here's an example. We have hydroxide. We have nitrogen. If they attract so much, negatively charged hydroxide, positively charged nitrogen, our first instinct might be to try to attempt to draw a bond, an arrow between the lone pair on this hydroxide and the positive charge on the nitrogen. that would be wrong, wrong, and wrong. It would be totally wrong. And the problem is that this is wrong at every level. The result here would be a fifth bond to nitrogen. And nitrogen, being in the first row of the periodic table, cannot possibly handle such a large number of bonds. Remember, first row of the periodic table, carbon, nitrogen, oxygen, cannot handle more than 8 electrons around the atom. Okay, that's 4 bonds. 5 bonds, totally wrong. Okay, another big problem with this that infuriates me is notice the arrow starting on the negative charge and moving to the positive. Okay, that's wrong too, because again, arrows are supposed to depict overlap of orbitals. I'm getting a little ahead of myself. Okay, here's the correct way to do it. The correct way to do this is to show hydroxide attacking the carbon and displacing the positively charged nitrogen in an SN2 reaction. Okay, so this opposites attract business only carries us so far. Okay, so that's our first problem. Is that this charge-charge interactions is very rare to provide an operative mechanism in organic chemistry, and for that matter, in bioorganic chemistry. Really, charge-charge interactions are very important for non-covalent bonding, not so important for covalent bonding, and in fact, potentially very, very misleading. So, cautionary note. Instead, we need to turn to molecular orbital theory. Molecular orbital theory can explain the otherwise unexplained, and I'll give you one example of this before we go back to our canonical example that I showed you earlier. Okay, so for example, this methyl ester has a preference for the syn conformation versus the anti-conformation. And to a first approximation, this should strike you as rather odd, right? Because in this case over here, the methyl group is as far as can be away from the lone pairs that populate the oxygen, right? Those lone pairs that stick up like Mickey Mouse ears above the oxygen. And so this anti-conformation should, to a first approximation, appear to be the preferred orientation. But you know, when we look closely at this, and we can using various spectroscopic techniques, what we find is actually the dominant conformation is the syn conformation. And you can start to understand this if you think about overlap of molecular orbitals. Okay, here is, here's again, that's the syn conformation, should appear to have some steric clash. But again, molecular orbitals explain why it is that it doesn't prefer that. Okay, so I keep talking about molecular orbitals. I think it's time for us to dive right in and start to dissect them and look at them in greater detail. And let's get started. So in molecular orbital theory, we're going to be talking about atomic orbitals. So the atoms of a molecule each have an atomic orbital associated with it. Okay, so the nitrogen has some atomic orbital. The oxygen, the carbon, even the hydrogen has some little tiny molecular orbital or some little tiny atomic orbital associated with it. Those atomic orbitals are found in S, P, D, and F orbitals. Okay, that's where the electrons hang out. They hang out in shells or orbitals. I prefer the word orbital, which describe their orbit as they orbit around the nucleus of the atom. Okay, and remember, those electrons, that's the business end of the atom. That's what endows it with functionality. That's what makes molecules the way they are. Okay, now, here's the thing. Oftentimes, these electrons are not simply in either an s orbital or a p orbital. Instead, they typically hybridize into hybrids of s-p orbitals. Okay? And we're used to this concept. These hybrid atomic orbitals are given the names sp3, sp2, and sp. Here's the important part. Okay, so this is review. I know that you've seen these hybrid atomic orbitals before. This is the part that matters to us as chemical biologists and bioorganic chemists. The S character of these hybrid atomic orbitals determines its stability. This totally makes sense. Okay, so an s orbital is a sphere, where in the very center of the sphere is the nucleus of the atom. Nucleus is positively charged. The sphere defines the orbit of the electrons. And in this sphere, those electrons can cozy up as close as possible to the positively charged nucleus. Okay, so this is a great example of opposites attract, and that attraction equals stability. On the other hand, a p orbital, as depicted up here, has the nucleus at a node between the two lobes of the orbital. Okay, so the nucleus is right here in the center again, but that happens to be a zone of exclusion where the electrons are not allowed to exist. Rather, the electrons in this orbital are hanging around either in this node up here or this other node down here. They're not in, or sorry, lobe. This lobe up here or this other lobe up here, they are not allowed to get up too close to the positively charged nucleus. And so for this reason, the S character of a hybrid atomic orbital determines the stability of that orbital, okay, of the electrons in that orbital. Conversely, the P character defines the instability. It defines how reactive and how nucleophilic those electrons in that hybrid atomic orbital really are. Okay? That's kind of like, defining how unhappy the electrons are. Okay? Happy electrons are found in these spherical s orbitals. Unhappy electrons are found in p orbitals. And what happens when electrons are in unhappy situations? Well, they will move. They will do everything they can to find more stable orbitals for themselves. Okay, so these are the atomic orbitals, specifically the hybrid atomic orbitals over here. And P character confers reactivity and basicity. So for example, if we look at a series of lone pairs found on carbon, what we find is that the higher the P character, the more reactive that resultant lone pair will be. Okay, and this can be dramatically illustrated in terms of basicity. Okay, so here's a lone pair in an sp3 hybridized orbital. Its pK is 50. Compare that against a lone pair in an sp or an sp2 hybridized orbital. The difference here is truly dramatic. Okay, so the pKa is only 41 in the case of the sp2 hybridized orbital, and then it's way down at 24 down in the sp hybridized orbital. This is an enormous difference. Okay, remember pKa's are a log scale. So in other words, this guy up here is 10 to the 26 times more reactive than this guy down here. And by more reactive, I mean how avidly it's going to be reaching out and ripping hydrogens, ripping protons off of its neighbors. Okay, and this tells us almost immediately that, for example, organometallic compounds are going to be extremely avid at grabbing protons to the point where they're incredibly flammable and nearly explosive. Okay, now this 10 to the 26 times again is huge, right? That's a 1 followed by 26 zeros. It's such a large number, it's hard actually for us to even imagine it. Okay, so enormous differences determined by this P character, S character. I hope by now everyone who's listening to this and everyone in my class can explain why it is that these guys are so much more reactive than these guys. And it should make sense just from geometric considerations as depicted here. Now, these hybrid atomic orbitals recombine into molecular orbitals in molecules. Okay, so the hybrid atomic orbitals only carry us so far. More often, these hybrid atomic orbitals are shared between atoms, and that sharing is what gives us bonds. Okay? Now, these molecular orbitals are given the names sigma, pi, and n. Okay? So these hybrid atomic orbitals form bonds with other atoms, and that yields molecular orbitals. The energy of these molecular orbitals is defined very specifically. And there's no way around this. I basically just have to tell you, I'd like you to memorize this chart on this slide. Okay? So please memorize the order of this reactivity, where sigma molecular orbitals are lowest in energy, pi are higher in energy, n are even higher. Okay, so these are the filled molecular orbitals. These are molecular orbitals that have electrons in them. And these electrons are depicted by the up arrows and the down arrows. Okay, that's a convention that you've seen before. Okay, now sigma makes sense. Sigma are the molecular orbitals that define single bonds. S for single, S for sigma. Pi, this defines double bonds. And that's convenient, right? Pi looks kind of like a double bond. N, the electrons in n orbitals, are the lone pairs that are hanging out around the atoms. Okay? So when the n orbitals are present, those are going to be the highest occupied molecular orbitals. So almost immediately, that clues us in that we need to pay attention to those lone pairs. Okay? What about the unfilled molecular orbitals that we're going to encounter in chemical biology? These will be found in 3 molecular orbitals. And again, I need to ask you to memorize these, the order of the energies. Okay? The lowest in energy are p orbitals. P orbitals are exactly what I showed you a couple of slides ago. Okay, that's them, these over here. This is what a p orbital looks like. It has a lobe and another lobe down here. P orbitals we find when we look at carbocations. Okay, the empty hole that is the carbocation is a p orbital. Okay, the other electrons that surrounds the carbon, that surround the carbon of the carbocation, those other electrons are in an sp2 hybridized atomic orbital. So the remaining empty atomic orbital is a p orbital. Okay? So most of the time we don't really have carbocations. The reason for this is that they are extremely reactive, being so low in energy. And so for this reason in biology, we very, very rarely find carbocations. In week 8, I'm going to show you an exception to this. But for now, let's keep in mind that we're just not going to see these very much. And again, the reason is biology takes place in water, and carbocations react avidly with water. Pi star. This is the antibonding complement partner to the pi orbital. And sigma star is the antibonding complement to the sigma orbital. And again, we're seeing this relationship where pi star is lower in energy than sigma star. Okay, so here's what I need to tell you. Good news, you don't have to worry about where all those electrons are in a molecule. And this is really fabulous news. Okay, if you just stop, take a moment, take a deep breath, pause, and appreciate this. Because the molecules we talk about when we talk about biology are fiendishly, fiendishly complicated. Okay? This goes back to the business that I talked about earlier of the hundreds, if not thousands, of pi bonds, the thousands upon thousands of lone pairs. The good news is we get to simplify all of that complexity down to just worrying about the frontier orbitals. Okay? So in other words, we only have to worry about the frontier highest occupied molecular orbital and the frontier lowest unoccupied molecular orbital. Okay? In other words, all we have to do is focus in on the highest occupied lone pair, highest occupied molecular orbital that has a lone pair in this n orbital, and also the lowest unoccupied molecular orbital over here. So in other words, if there's an available p orbital, it's going to react first. Okay, if there's a carbocation, everything else will come to a halt, and carbocation gets its day in the sun. It gets to dance around. Okay, if there is a lone pair will be the dominant reactivity. Okay? This is good news. Okay, it simplifies everything. We just have to look for the highest energy HOMO and the lowest energy LUMO. Okay, what does this mean? What this means is that this highest occupied HOMO is the filled frontier orbital. And this is the orbital from whence all nucleophilicity, all basicity springs forth. Okay? And I apologize for the kind of antiquated English, but really that's how I think about things. Okay? This is the orbital that is the business end of this complicated molecule. It doesn't matter how many possible lone pairs it has. It doesn't matter how many different possible configurations it has. All that really matters is its highest high and its lowest low. Okay? Again, this is majorly important because it simplifies things for us. Okay, so this HOMO, highest occupied molecular orbital, is the filled frontier orbital, and it's a nucleophile in reactivity. Okay, now, the intrinsic nucleophilicity is governed by the energies of these molecular orbitals, where again, the highest in energy is the N-bonding, the non-bonding molecular orbital that has the lone pair, and the lowest in energy are the electrons of the sigma or single bonds. Okay? To reduce it down to simplest terms, we're never going to be really seeing reactions that start with sigma bonds. Okay? It just doesn't happen in chemical biology. Most of our reactions are going to spring forth, are going to spring from lone pairs that are in non-bonding orbitals, occasionally electrons and pi bonds. But really, we don't really have to worry about the electrons and sigma bonds. We know they're there. You know they're there. They're there. But we don't have to get wrapped up in them. And this, again, is good news because there's a huge number of electrons in these complicated molecules that have, thousands upon thousands of atoms. OK, what about the lowest energy unoccupied molecular orbital, or the LUMO? This is the unfilled frontier orbital. And the lowest energy unoccupied is the most available molecular orbital. This is the molecular orbital that's going to be the center of attention for reactivity. Okay, and again, where you have these complex molecules, this is kind of like the funnel to which all reactivity zooms towards. Okay, now again, we need to know this order over here, where P is lower in energy than pi star, which in turn is lower in energy than sigma star. So if we're given a choice of different sites for nucleophile to attack, nucleophile will choose every time to attack the p orbital because it's lowest in energy. And again, we see p orbitals when we look at carbocations. If there are no carbocations present, which again, like I said earlier, is exceptionally common because carbocations are very, very rare in biology, where biology takes place in water. So if there are no carbocations present, we can eliminate this one, and we start focusing on pi star orbitals. If there are pi star orbitals that are available for reaction, then it's likely that this will be the dominant reaction. Occasionally, you come across a molecule that doesn't have a pi star, in which case then you might have an attack on a sigma star. This is rare, okay, especially as depicted here. This is utterly wrong as depicted in this slide. I find it offensive, but I'm stuck with it. This might happen, for example, if there was a sulfur here. Then you might have this sort of reaction taking place. For now, let's keep in mind that we're going to probably be having reactions. Our electrophiles in our reactions are going to be molecular orbitals consisting of antibonding pi bonds. Okay, so it's the pi star or antibonding pi molecular orbital. Okay, I want to switch gears. If you have any questions about molecular orbitals or hybrid atomic orbitals, don't hesitate to shoot me an email or talk to the TAs, come to my office hours, etc. We now have to talk. I've told you about what you do to decide what the reaction mechanism is. We now have to talk about how to actually tell me what that reaction mechanism is. Okay, so oftentimes in chemistry, we have some notion that molecules are reacting, but we need a clear way of communicating that reactivity. So organic chemists have developed this wonderful vocabulary using arrows. And so let's take a closer look at what those arrows are. The arrows are going to be starting from highest energy occupied molecular orbitals, the HOMOs. And they're going to be ending on the lowest energy unoccupied molecular orbital, the LUMO. Okay, this is a golden rule. This is a rule that always applies. Okay, your arrows start on orbitals, they end on orbitals. They start on HOMOs, they end on LUMOs. And again, they're always going to start on the highest HOMO and the lowest LUMO, and they'll end on the lowest LUMO. Okay, so again, that lowest LUMO is the lowest energy unoccupied molecular orbital. That's the most available, and in turn, that's the most reactive. Now, the problem is, again, we oftentimes have many HOMOs, we have many LUMOs. What's an organic chemist to do? What's a student supposed to do? So when in doubt, refer to this idea of looking for the highest HOMO and the lowest LUMO. I can simplify it, cut it down to make it even easier for you. Most of the time, just put your pen on a lone pair and start pushing electrons to end on the best electrophile. Okay, it's that easy. If you're in doubt, you're stuck there at your desk during an exam, you don't know where to start, put the pen on the lone pair and just start drawing. Okay? End the arrow on the best electrophile. 9 times out of 10, 99 times out of 100, maybe even more, you'll get the answer right just by doing that. Okay, so I need to talk to you about some rules. We have rules because this is a language, and in order for us to be clear in what it is that we're communicating, we need to have some conventions. Okay, the conventions we're going to follow in this course are the following. And by the way, before I present these conventions, I should tell you, I'm a stickler for these rules. Okay, if you give me something that doesn't follow these 3 rules, chances are, even if it's correct conceptually, it won't get full credit. Okay, and the reason for this is it's kind of like turning in an essay that has incorrect grammar to your English class or something like that. What's your English professor going to do? You know, give you an A for great ideas and a C for bad English? No, your professor is probably going to give you a C overall because the goal is to communicate effectively. Okay, so in the same way, when we speak using the language of arrows, we have to follow these conventions because this is what convinces us that we know what we're talking about. Okay, so the conventions are arrows never indicate the motion of atoms. And this is one that, if we stop and think about it, actually is kind of profound. I think that all of us are used to having arrows showing, you know, football player who's over here, let's say the quarterback, moves back here and then gets behind this guy, and then another arrow shows this guy moving forward. Those are the kind of arrows that you've been drawing, you know, I guess since you were able to draw arrows, okay? Which is to show motion, to show the 4th dimension, really, to show some element of time. Organic chemistry, we don't use arrows in that way. Rather, we're using arrows to depict overlap of orbitals. We're not depicting it in terms of time, we're depicting it in terms of thermodynamics, not kinetics. In other words, we're depicting an overlap of orbitals that's allowed. Okay? So arrows do not indicate the motion of atoms. Yes, it's true that atoms must cozy up to each other. That's kind of understood, that's lurking in the background, but that's not really what arrows show you. Arrows never start or end on charge. Since these arrows are depicting the interaction of filled and unfilled molecular orbitals, charge is irrelevant. Okay? Charge, formal charge, is one of those nice conventions that makes Lewis structures so much easier to understand. Yet the charge itself does not show you where the electrons are. It doesn't show you anything about the molecular orbital. And so drawing an arrow from one formal charge to another is worthless. Okay? So again, arrows never start or end on charge. Instead, here's the one that really embodies everything. arrows begin with lone pairs, with pi bonds or sigma bonds, and end on unfilled orbitals. Okay, and I want you to be really precise about how you draw these things. That precision indicates that you understand what is going on. Okay, and drawing your arrows to precisely end where it is that that pi star orbital should be, you're telling me something. You're telling me a story. You're telling me where it is that those electrons are going to appear, and in doing that, you're describing to me the reaction that's taking place. Okay, so I need to have all of these things taken care of when it comes time to for exams and things like that. Okay, makes sense? Okay, so why don't we take a look at an example? Okay, so an example is this very simple problem. Okay, and the problem is we're going to have a lone pair on the nitrogen over here, and we're going to do a simple substituted nucleophilic attack, substituted nucleophilic reaction that substitutes for chlorine this lone pair on nitrogen, or this nitrogen, this amine. Everything looks good. This is a reaction very similar to the one I showed you at the very beginning of the class. Avoid the poisoned candied apple of simplicity. Rather, fall back on HOMOs and LUMOs. Let me show you what I mean. Okay, and to do that, I need to roll up the screen. Here's what I mean by avoiding that tempting poison apple of simplicity. Okay. So, in this example, there's a lone pair on the nitrogen. All right. Okay, so here's our reaction again. And the simple, and I would call it even simple-minded possibility, is for the nitrogen to simply displace the chloride as an SN2 reaction. All right, so in the simple case, we have this reaction mechanism here. Okay. Okay, this case I would call an SN2 reaction, right? Substituted nucleophilic 2 reaction, and this will give us this guy over here, and then this can lose a proton. Okay, so I'll show this base. The base, for example, could be chloride. Wow. Okay, and this base can deprotonate this nitrogen, giving us the product. Okay, now what's wrong with this? This is totally, totally wrong and completely unacceptable. It appalls me. It's upsetting to me. What's so wrong about this and so appalling is the fact that we're attacking a sigma star orbital. Okay, this is an attack on this sigma star orbital over here, right? When we have several perfectly good pi star orbitals that are available, right? So sigma star is not the lowest energy unoccupied molecular orbital. Far from it. There's plenty of pi star molecular orbitals that are going to be lower in energy. Why don't we explore those as a possible reaction mechanism? Okay, so a different mechanism would start. Okay, so I'll draw some lines through here. A different and more correct mechanism will this time have the lone pair in that N non-bonding orbital of nitrogen attacking the pi star orbital of this alpha beta unsaturated carbonyl. Let me show you. Okay, so here's the lone pair. It's now going to attack the pi star molecular orbital. Electrons bounce, bounce, all the way to the electronegative oxygen. Okay, so again, this is attack not on a sigma star, but it's attack on a pi star molecular orbital, an antibonding orbital. Okay, now why is this so much better? This is better because the pi star is lower in energy than sigma star. And so for this reason, this addition-elimination reaction is greatly preferred. Okay, this is actually the operative mechanism for this reaction. Okay. You could continue on. I encourage you to do so. And in the end, you get this product over here. Okay, there will be many times in this class I'll kind of set stuff up for you. I'm going to then let you finish that off on your own. I apologize for that. This is upper division organic chemistry, upper division chemical biology. We're at that point where I don't have to show you every step. There are fundamental steps that I want you to know. There are fundamental steps that I expect you to know, but I'm not going to show them to you during every lecture. Okay? Rather, I want you to go home. I want you to fill them in your notes. I want to make sure that you know them, because on the exam, I will ask you to show me those steps. But on the other hand, I'm not going to dwell on them today. Okay? I just don't have enough time to talk about them in a class of this length. Okay, so the lesson from this is clear. The lesson is don't be tempted by simplicity. Instead, look at the overlap of orbitals. Which one is a better overlap? Overlap with the sigma star or overlap with the pi star? Pi star is lower in energy, and it's therefore greatly preferred. Okay, let's move on. I have to lower this again. All right. Now, the other thing to make this work is that you also have to make sure that you're drawing the correct Lewis acid structure. For the most part, I don't think this is going to be a problem in Chem 128, but it is important that you set things up correctly. Okay, if you are drawing, for example, 5 bonds to nitrogen, a lot of the reactivity of this N oxide is not going to be apparent because this is totally wrong. Okay, similarly, you know, in terms of the number of bonds that you draw, this helps you in terms of keeping track of things. For that matter, it's also essential for you to depict correctly the formal charge. Oh, thanks. Sorry. This formal charge helps to guide us. For example, the negative charge on this carbon over here, that should look kind of funny to you, right? Carbions, that should look funny. That should be extremely reactive. So formal charge helps to guide us in terms of drawing these correct mechanisms. I'll have a lot more to say about hydrogen bonds. I don't really care about dative bonds. We won't see them in this class. Let's not get into it today. I'll talk to you more about hydrogen bonds in a moment. Okay, so arrows start with bonds or lone pairs. And here are some correct depictions of arrows. Okay, so in this case, where we're showing a bromide leaving for an elimination reaction, the bromide takes off. And notice that the arrow is starting at the carbon-bromine bond. Okay, in other words, the electrons in that carbon-bromine bond decide to step out the door and leave with their friend, the bromine, giving us a bromide ion. Okay, so here's electrons that are starting with another bond, in this case a pi bond. Here they are starting with a sigma bond, here they are starting with a pi bond, and here they are starting with a non-bonding lone pair. Okay, all 3 of these cases are correct. Contrast that with these cases over here where I'm showing you arrows starting on charges. This again is deeply appalling and totally wrong. So arrows do not start on atoms. So for example, like this or like that, Instead, we want to draw them starting on the bonds themselves. This should make sense, right? The arrows are trying to depict the overlap of orbitals. They need to start where the electrons are. The electrons are found in these bonds. Electrons are not found in this negative charge. They're not really found around this bromide. Instead, we're talking about the electrons that are shared between bromine and carbon. Those are the electrons that matter. Okay, now, that's where they should start. Let's talk about where they should end. So arrows need to end on atoms and bonds. Okay, so here's a lone pair attacking a proton. It's ending directly on that proton. Okay, so here's it ending on atoms, the proton. Here it is ending on the carbon of a carbocation. Here it is ending on the hydrogen or the proton during a beta elimination step. Okay, so atoms never terminate in empty space. So for example, when bromine is stepping out the door, the electrons don't simply hop out and then go over into empty space. For that matter, this arrow would be wrong if it started at this carbon-bromine and then had the electrons just going off into empty space. That's not correct. The electrons don't get to walk off into empty space. That would be extremely high in energy and extremely repellent. Rather, the electrons get to end on this bromine atom, giving us bromide. Okay? So arrows need to end on atoms. They will depict, again, this overlap of filled and unfilled orbitals. Okay. Hydrogen, for that matter, is always attached to something. Okay, I'm starting to get down to my pet peeves, but this is one of those pet peeves that does matter. Okay? Hydrogen is not some atom that kind of like is floating around next to the molecule. Rather, hydrogen is directly attached to some particular atom. And this matters a great deal because where it's attached will determine to a large extent whether or not it's going to be acting as an acidic proton or perhaps not acidic at all. Okay, so these terms, proton, hydride, and hydrogen atoms are three different depictions of the hydrogen atom, and they have three different meanings. They have totally different meanings. Okay, so H plus is the proton, H minus is the hydride, and H radical is the hydrogen atom. We don't find them just kind of floating around like this in the chemistry that takes place inside cells. Okay, protons aren't just floating around inside the cell. Rather, they are always attached to something. Maybe they're attached to a water molecule to give you a hydronium ion. But they're not just kind of hanging out. They're doing something. Okay? Hydrogens do not like being by themselves. Okay? So in other words, what you want to avoid is showing a proton just kind of hanging out in space, waiting around for some lone pair electrons to attack it. That's not what happens. Hydrogen doesn't get to do that. Okay? Furthermore, hydrogen radical also doesn't really occur, nor does hydride really occur. Okay? Rather, in solution chemistry, we find species that can either donate a proton, donate a hydrogen radical, or donate a hydride. Okay? So what I propose you do is instead of depicting H plus as a reagent, instead depict H plus as catalytic quote unquote H plus. Those quote unquotes are going to tell us that yes, we mean H plus, but what we really mean is we mean H plus that's been picked up and delivered by some other species. In this case, that might mean attached to this methanol molecule that's going to be its delivery character. Or you can even write catalytic HA, where in this case it's HA that's attached to the conjugate base that's going to be delivering the proton. Okay? Any one of those is fine. It is important for you, however, to follow these conventions because they communicate to me that you know what molecular orbitals are being overlapped, and it tells me whether or not, you understand the chemistry that's involved with these reactions. Okay, one second. Okay, I want to conclude today's lecture by discussing with you one other element of hydrogen atoms, and that's the hydrogen bond. Everyone needs a favorite bond. My favorite bond is, of course, the great Sean Connery. But today I'm going to be talking to you about a second favorite, which is the hydrogen bond. Okay, the hydrogen bond governs so much of biology that it is essential for us to really get to understand it correctly. Okay, so hydrogen bonds are actually largely a Coulombic interaction. They describe the sharing of a hydrogen atom between two partners. One partner is going to be our hydrogen bond donor, and a second partner will be a hydrogen bond acceptor. And this hydrogen bond will be depicted by this dashed line. Okay, so this dashed line is going to be our convention for hydrogen bond. We're going to use this a lot. Okay, hydrogen bonds, for example, hold together the two strands of DNA. They make molecular recognition possible, the non-covalent interactions between molecules. So hydrogen bonds, absolutely essential to chemical biology. However, it turns out that the energy of the hydrogen bond is very sensitive to the environment and the geometry that's involved with the sharing of that hydrogen. The geometry, in this case that I'm showing you over here, is of a perfectly linear hydrogen bond, which is the best possible example. Okay? So in this case, the lone pair on this water, on this oxygen of water down here, is perfectly positioned to share this hydrogen bond, this hydrogen of this water up here, and the oxygen, hydrogen and oxygen are lined up as a straight line. Oftentimes that isn't the case. Okay, so for example, we can look at hydrogen bonds that are found in the active sites of enzymes, and we find instead of having this neat straight line, we get a bendy line instead. That bendy line, that bent hydrogen bond, much, much weaker. Okay, so this is kind of the optimal geometry. Optimal hydrogen bond acceptor, which is a lone pair, optimal hydrogen bond donor over here. And this will be our canonical hydrogen bond. Now, here's one of the problems. One of the problems, amongst others, is curved arrows. Curved arrows confound us when it comes time to talk about hydrogen bonds. The reason is curved arrows and hydrogen bonds simply don't mix. Curved arrows depict the overlap of filled and unfilled molecular orbitals, whereas hydrogen bonds are showing us a partnership of sorts between a donor and an acceptor, and there really isn't this sort of overlap that leads to a covalent bond in the case of hydrogen bond. And this becomes tremendously confounding. So for example, if you wanted to show transfer of the proton on this nitrogen atom to the lone pair of the oxygen, you might be tempted to simply draw a hydrogen bond in here, and that would be utterly incorrect, because this hydrogen bond is basically saying the hydrogen is somewhere between here and there, somewhere in the middle, somewhere on the sides. Whereas over here in the case of the curly arrows, you're saying, no, it's going to pick this up. It's going to pick up the proton wholesale, hang on to it for a while, and give you a positive charge on oxygen. These are two very different depictions. So what we were going to be doing in this class is showing those hydrogen transfers as an explicit step. Okay, so hydrogen bonds are going to be useful for us for talking about non-covalent interactions, but not useful at all for talking about covalent interactions, the reactivity that I've been talking to you about today. Turns out that hydrogen bonds, that proton transfers of the sort that I showed in the previous slide, these sorts of proton transfers over here, are extraordinarily fast. Okay, they're oftentimes diffusion controlled. In other words, they hit the speed limit of reactivity for reactions that take place in solution. That kind of speed limit and that kind of proton transfer ability is actually tremendously useful. Okay, so this is a diffusion-controlled reactions. So proton transfers to and from heteroatoms, very, very fast. Proton transfers, for that matter, in the same way that hydrogen bonds require linear geometry, proton transfers also require linear geometries. And I can tell you that almost immediately this is going to annoy you. This takes away one of the conventions that you mislearned back in sophomore organic chemistry. I know it was cool back then to show a proton transfer as a neighboring oxygen, say, picking up a proton over here on the nitrogen, and you have this completely ridiculous and totally crazy four-membered ring transition state. It galls me to even say this. Can you imagine four atoms getting together to form, some sort of very strained four-atom ring transition state? It's total insanity. Even more insane, notice that the geometry between oxygen, hydrogen, and nitrogen is not perfectly linear. Instead, it's bent at a 90-degree angle. And these kind of proton transfers do not happen this way. Instead, they exclusively prefer a linear geometry. So only linear geometries are going to count when we talk about proton transfers. And so for this reason, I need to take this, this particular step out of your vocabulary. Okay, now it was acceptable back in sophomore organic chemistry. It's no longer acceptable. So acids and bases are required to catalyze proton transfers into tautomerizations. So instead of showing it like this, a much better alternative would, not even alternative, the correct way to depict this would be to show the oxygen picking up a proton from a catalytic acid, and then in turn the conjugate base of this acid acts as a base to deprotonate the neighboring positively charged nitrogen, the ammonium ion. Okay, so in all cases, we're going to see that acids and bases are required to catalyze these proton transfers. And that turns out to be a general rule. And the good news for us in terms of chemical biology is that oftentimes, or in all times, we can find abundant numbers of different molecules that are all too willing to volunteer to be those catalytic acids and bases. And the obvious examples would be, for example, water. Water can be a hydronium ion to act as a proton donor. Water can also act as a base to accept protons and become a hydronium ion. And since all biology takes place in water, feel free to use water as that catalytic acid and that catalytic base. Okay, we've come quite a ways. I've shown you proton transfers. I've shown you how to draw arrows. We've talked about the rules that govern these electron transfers in terms of filled, unfilled overlap of molecular orbitals. We're now going to transition to looking at some examples of this. And I'm going to show you this on Thursday when we talk about the molecules found on Earth that compose all living things. So why don't we stop here? When we come back next time, I'll be showing you examples that apply the principles that we've talked about today. Thank you very much.